Please be welcome. My name is Nicolaas van der Voort, and today's webinar will take a wholesome view of colocalization. So we'll be looking at acquisition, we'll be looking at quality control, colocalization, pitfalls, and restoration. And then finally, we'll be looking at the analysis options that we have when we want to look at colocalization. Now, because of this, I will try to cover all these topics just to paint you a complete picture. And as a consequence, we'll need about 30 minutes for the presentation and 15 minutes for a demonstration. It's a bit longer than what you used from us, so apologies for that. If you need to leave early, we make the recording available so you can catch up if you need to. Right, so the immediate reason for this webinar series is a recent paper that we published in Microscopy Today. And when we started with this series, we actually realized that there is a lot of material to cover. And so you guys can see my, yes, you guys can see my screen. So we actually realized that we have material not for one webinar, but for a series of webinars. And so in this first webinar, we'll sort of cover the whole picture, but we won't have time to go into too much detail. Then in the second webinar, which we plan to have in September, and the date will be announced, we'll go more into the coefficients. Which coefficients should I use? What analysis method should I use? When? What threshold should I use? All these questions will be covered in session number 2. Then in session number 3, we'll go more in depth in the quality control, colocalization, more advanced types of correction needed. And finally, in session number 4, which is planned in October, we'll cover some advanced topics that include threat, colocalization on big data, and colocalization on super resolution methods as well. So STAT on one hand, but SMLN and MinFlux on the other hand. Okay, so like I mentioned, we'll be going from acquisition to restoration and colocalization pitfalls to analysis. And so when you start a colocalization experiment, you start out with some experimental goal in mind, right? You typically you want to learn something about the biological system that you're studying. Most of it is biology anyway. And so you have in mind what you want to know. And then from that follow the experimental goals. So you make your sample, you stain your sample, and you maybe have your cells and cell cultures, or you're growing organoids, or you have another type of tissue. And then when you do your imaging experiment, it's a well-known trade-off that you have to leverage, or you have to somehow make a compromise with respect to what part of the imaging pyramid you are optimizing. So for example, if you're interested in some live process in a live cell, you want to either measure very fast or for longer periods. But if you try to do that at a high SNR, you get a lot of bleaching quick, so you're sort of forced to go to a low SNR, and you have to make a compromise there. On the other hand, if you don't care about temporal resolution because your sample is fixed anyway, then you can just go for a high SNR from your image and then go for the high SNR. So Huygens, of course, does deconvolution. And what deconvolution does is it makes your imaging pyramid bigger. So you don't have to compromise as much, or you have more room to compromise in a way that that is best aligned with your experimental goals. So, for example, if you're doing a live experiment, then the deconvolution is very good in transforming low SNR data into high SNR restored data. And when you leverage that correctly, you can dramatically extend either the duration of your live experiment or, as your scanning system or camera system allows it, measure a lot faster than you would otherwise be able to do. Now, on the other hand, if we're interested in getting high resolution, because maybe my sample is fixed, then you just measure a high SNR image to start with, and then the deconvolution will add much more resolution. So you'll be able to see more detail. So you can leverage, you can turn the knob, and you should start thinking about that already when you're before you start imaging. Another very important aspect is the Nyquist sampling. So of course, many of you, I think all of you are already familiar with this. Just if you sample according to Nyquist, you will get most resolution out of your image. If you undersample, you will have less detail, so less resolution, and that can obscure actually important detail. So this picture shows an image that we'll be looking at more today. And what you see is it's a cell undergoing mitosis, and the red is the chromatin bridge and the green is the kinetic core complex. So the chromosomes are all rolled up, and they are about to be pulled apart. The two copies of the DNA for each chromosome, and yeah, that's the detail that you can see here. And this one has already been pulled apart. And what you can see, so for increasingly bigger pixels, of course, the resolution gets worse. I see less detail, but you can also see the Pearson coefficient as a measure for colocalization going up. And that is not because the biology is changing, just your resolution is getting worse, and that's causing objects that are really separate to have a bigger resolution. They start to overlap. That makes the Pearson coefficient rise, but it's not an accurate measurement. So we'd rather have a higher resolution if we can. So use the Nyquist calculator. We have an online tool available. It's even available as an app on your phone for Android. And if you have an Apple phone, then you can just use our online tool. And so for confocal, it's typically 50 nanometers. I know that, and I'd just like to mention that we've heard some acquisition software have a button, use Nyquist. And unfortunately, so in some cases, it is the correct Nyquist, but in some cases, the pixels are too big. I don't know why they implemented it like this. The Nyquist rate has been established really firmly for a long time. So if you use the Nyquist rate from our app, then you will get the best resolution you can. Okay, so let's have a closer look at colocalization pitfalls. What can happen during acquisition and how can we correct for it? And we'll be going through also the options that are available in Huygens to correct for this. So let me start with noise. And noise is most prevalent for scanning-based microscopy methods, confocal notably, but also STAT, of multiphoton, of course, array detector, ARIScan as well. And so yeah, what you see here is, suppose you have two noise realizations here of the same object, but just because it's noisy, then here they seem to not overlap, but that's just because of the noise. So the noise makes the Pearson coefficient go down. And here we show an example. This is an experiment we did with Lucas Kapitein from Utrecht. And here we did an example where we measured every 10 seconds 10 Z slices. And at first, the intensity is good enough to just analyze the raw data, but it just bleaches so fast, and the sample is not happy at all. And we're not able to acquire a long time series. Deconvolution makes it a bit better, but what's a better approach is to start with low SNR data. And this is what we show here. So here we dramatically decrease the amount of irradiance that we use. So lower integration times, lower laser intensities. And yeah, as you can see, over the course of tens of minutes, the intensity just drops very slowly. And each time using a deconvolution, we can get a very high SNR deconvolved result out of that. So if you have noisy data, deconvolution is a great counter. Other microscopy types, they suffer from blur. So wide field is the most notorious, of course, for having a lot of blur, but also other camera-based methods. So you can have, for example, if you have a spinning disk type microscope, then and your sample is a little bit thicker, you will also have blur. You may not notice it immediately, but it is there, and that will also deteriorate your image quality. And for light sheet, it can happen as well, although the story is a little bit more complex there. So in any case, what that does is it creates an overestimation of your colocalization. So here for this raw image, Pearson coefficient is 0.89, so 90%. And that's just because all of the blur everywhere is co-localizing, and that's just really the majority of your signal. And if you make a representation of that, you can see, okay, where's my co-localization happening? You can see it's happening here, which isn't very good. It is not specific type of co-localization. So what you can try to do to counter that is you can try to sort of increase the threshold that you use to threshold your data to say I only count it when it is higher than so and so much. But and you can see it increases a little bit here, but then here at the sides, all these places start to drop below the threshold. So that's also not a solution, of course. So when you do the deconvolution, it's really great for deep blurring. And you can be, this is wide field data, you can just get also really fantastic 3D information on the wide field data. You can look at, here you can see that all these individual kinetocores are co-localizing with the chromatin bridges in 3D, and just very effective for deep blurring. Now, so noise and blur will always be there, essentially. If another issue that is nearly always also there is chromatic aberration. And that's because just the glass that the lenses are made of have a different refractive index for the blue light and for the red light. So the blue light is typically focused more strongly, and the red light is focused more higher in Z. And you can get objectives to correct for that, and those can be quite expensive. But that is not a perfect solution because the correction typically works for one setting. So one cover glass thickness, certain working distance, certain embedding medium. And if any of these things changes, so you're not using water, but you're using maybe water with a lot of protein in it, or your cover glass thickness isn't very precise, it's not 170, but it is 180, then these effects can return. So if you just do the colocalization correction in Huygens, then you can correct for it like so. And that is just a very robust way to deal with it. And I'll not go into detail, but I'll just briefly mention you can do this with a bead measurement or without. And if you're doing it without, then you're just aligning the channels such that they match to the best possible configuration, which is what you want in the far majority of cases. So beads are optional, you don't need them, but you can use them. Moving on, crosstalk is of course also a known phenomena. It's not always present, but it leads to an overestimation of the amount of colocalization, right? So if the fluorophore of the signal from fluorophore A has a bit of a wide spectrum, therefore also appears in the channel B, then the signal appears at the same place in both channels, so that appears to be colocalization, but it's really crosstalk. So if you have crosstalk, you need to correct for that. And we have also a very good tool to do that. And we'll be going into that, how to detect it accurately, how to do it for maybe just a few channels, how to do it for many channels, how to get rid of autofluorescence. We'll cover that in session number 3. Here I just show an example. So this is a hoogst that is bleeding through crosstalking into the mitochondria channel. And yeah, you can see in the line profile that just in the mitochondrial channel, there's this offset here. Now, if we correct for it, we can see that the signal in the nuclear channel increases and the signal in the mitochondrial channel decreases, right? Because it's not supposed to be in the mitochondrial channel, it's supposed to be in the nuclear channel. But here at the edge, you can just see that there is this signal in the mitochondrial channel, which is actually in the mitochondrial channel. So what's happening here is that you have a nucleus and a mitochondrion that's just sitting at the bottom, and then they appear to overlap. So this signal is supposed to be there and that is maintained. So you only correct for the contribution that's due to crosstalk and not for other contributions. Okay, so then we have some miscellaneous issues such as hot pixels. If you have a camera, they age. You can get hot pixels in your data, and that's just a nuisance. It degrades your image quality overall. So we have a correction tool for that, very user-friendly. And finally, you can have bleaching, especially if you have time series, but also if you have big Z series, then you can have bleaching, and that will make it more difficult to compare channels along time or in Z. So our bleaching correction tool makes sure that the intensity is of that the effect of the bleaching is countered over a time or Z, such that you can make a good comparison along those dimensions. Now, lastly, as an honorable mention, I'd like to mention unspecific staining. If you use antibodies and your secondary antibody doesn't uniquely recognize only the primary, but you have unspecific staining, maybe you get an image like this. I borrowed this from the internet from Mr. Anders Lunde, who posted it on ResearchGate. And yeah, so this will lead also to an increase of colocalization, so a false increase of colocalization. Because the unspecific staining is co-localizing probably, and that's not what you want. So unfortunately, Huygens cannot correct for the unspecific staining, and that is because um, Huygens cannot know what labels did you intend to be there and what labels did you not intend to be there. So Huygens does not have that information. So the best thing really is to solve this during sample preparation. And if you can't avoid it, then you can work around it. So what you can do is you can make a blank. And so you could, for example, only add a secondary antibody and not add a primary antibody. And then you would measure how much co-localization is due to the unspecific staining. So sort of establish a base level from which you can then measure deviations from the base level. Okay, so this is an overview again of the topics that we just covered in this short time period. And again, we have separate videos on these things to go into them in more detail. It's important to cover these in a certain order. And Huygens also assists you. So in the Huygens software, you will find it also repeated in this order. So you'll see hot and cold pixels you want to do first, then crosstalk, then deconvolution. Then you have the bleaching correction is incorporated into the deconvolution. Finally, you want to do the chromatic aberration correction. There's some cases in which you can use a different order, but this order just gives you good results in the majority of cases. And so it's important that you stick to this order unless you know good reason not to. Okay. Right, and all of these are also, so one important aspect of quality control is recognizing these issues. And for that, we also have our quality control tool. So the experienced user can maybe recognize these by eye, by experience. But if you have, maybe you're a bit newer, or your job is to train a lot of users, then we have the quality control tool, which automatically determines which issues are there, and then it asks the user, hey, I think this is there. Can you please verify this? You have to watch for this and this. There's information there. It's really useful to help you along. Okay, so at this point, we have a well-recorded, restored image. And now we'd like to analyze that. We're using colocalization. And it's worthwhile just to take a minute to take a little step back and to reconsider why colocalization is a very relevant thing to do. Well, so it allows you to measure the interaction between any type of biomolecules quantitatively. So Huygens really enables you to do this very quantitatively, very well, very accurately. So some application is, for example, if you want to measure interactions. So if you have some family of proteins that are working together in some way, that are creating some signaling pathway, then you can do live experiments, for example, and you can also fix them. You can see where the colocalization is happening. That's really the beauty of fluorescence microscopy. For example, if two proteins are interacting in the mitochondria or in the cytoplasm, all these things you can get out from your colocalization measurement. You can do condition testing. So you maybe have a wild type situation, and then you see what the effects are if you add a marker like interferon gamma or ATP, or you make a mutation in your protein and you measure all these things. And then you typically get a graph that looks like this. Or you can do time or concentration series where you either wait in time or you add an increase in concentration, a certain additive, and you look how the response changes. Maybe you try to fit some curve to it. So when considering these applications, so two things are I'd really like to point out. So the first thing is that provided that you did a good job of restoring your data, the Pearson coefficient will give you a good, reliable readout. There's many people there's many questions about what coefficient should I use. You can even flip it around a bit. If you did a good job on the restoration, you have performed a high quality deconvolution, you checked for any other effects that might be there, then that's really ensuring the quality of your measurement. And the Pearson coefficient is a perfectly fine indicator for how much colocalization you have. Now, the second thing I'd like to say about this is that you need a lot of statistics. So the rule is like you need three, or this is like a heuristic rule that a lot of people stick to. You need 3 independent experiments, 3 cell cultures if you use that on 3 different days, and each experiment at least 20 cells, something in that order. And so you need automation. And Huygens allows you to automate these things very well using the workflow processor. So the workflow processor is the tool that has this symbol. Essentially, you run you figure out how you want to set up your analysis, and then you run it on all your data, and you get the results out. Another big part of statistics is regions of interest. So typically, if you acquire an image, so let's talk about cell culture for the moment, then you have like a square image, and the cells do not necessarily stick to the frames of your square image, right? So you may have 1 or 2 cells that are completely in the image, and a third one which is sort of half included in the image. And it makes sense to do your analysis not per image, because the borders are sort of arbitrarily chosen, but to do it per cell instead. And so cell is a logical unit for biology. So then you can use the region of interest tool to just draw a region of interest around each of the cells, measure the colocalization for that. And on top of that, it helps you get more statistics. So I advise to do that. And it's not just about cells. So you can also look at, if you can look within a cell, you can look at how are things near the nucleus or more towards the periphery of the cell. You can look at organelle colocalization, like I was mentioning. If you have some stainer for the mitochondria, you can, for example, look at colocalization within and without the mitochondria. There's many different things you can do in this manner. Now, I mentioned before that Pearson coefficient is fine. There is one exception, and that is so. Let me clarify before let me clarify one thing. So you have Pearson, and then you have methods like Spearman and Object Spearman, and you have a whole family, but and they have different pros and cons. But for most cases, if you have Pearson, that will give you the biological readout that you want. Now, there's an exception where you'd rather like to use a coefficient like the Manders coefficient. And to illustrate that, I've drawn here two cases. So I have here in green something that might be an ER, and in red something that might be a certain protein, and that protein binds to the ER. So if I have this protein in a low concentration, then most of the proteins are bound, but the ER is mostly empty, right? So most of the binding places in the ER are not covered. So if I would measure the Pearson here, maybe I get 50% or something close to that. Whereas in this case, I now increase the concentration of my red protein, and then I get this all the binding pockets for the ER are filled. So now the red protein for a much higher fraction is in the cytoplasm, and the green but on the other hand, the ER is now more covered in red. So this case, there's more red not co-localizing, but there's more green co-localizing. So maybe for the Pearson coefficient, that sort of evens out. Maybe you get something close to 50%, and that's not a very accurate readout. But so what the Manders coefficient does, and there's similar coefficients, but we use Manders. You can have the concentration of A, so A and B. And here, so how much of green is covered with red is 10%. How much of red is covered by green? It's much higher, it's 90%. And here the picture is reversed. So to put that in a graph, and this is just an illustration, right? It's not data, it's just an illustration of how this pattern goes. So this Manders coefficient goes down, this goes up. So using Manders, I can really follow my biological phenomena in a lot of detail, whereas the Pearson coefficient sort of remains neutral. So in this case, adding the Manders coefficient just gives you a lot more insight into the biological background. Finally, there is object-based colocalization, and that is when, so you're no longer getting coefficients, but you're segmenting your data into objects, and if your objects make a lot of sense, then you can do object analysis, object-based colocalization. So in the example of the kinetochore complex with the chromatin bridges, each object represents one kinetochore that's attached to the chromosome. It's a very clear interpretation of your objects. And then you can look at object intersection. So is it intersecting with an object in the other channel? But you can also look at the object distance. So how close is the nearest object in the other channel, for example? And what the cool thing about that is well, you have object-based, you have a lot of statistics, and you can do multivariate analysis. Like I show here, then if the bridge is dissolved, then that population is located here in my 2D histogram. If it's still intact, it's located here. So I can do multivariate analysis. Now, I'd just like to give a brief overview, because many of you, of course, have Huygens licenses. Which option, which step that I've been talking about corresponds to which option? So the hot and cold pixel corrector is included in the basics. So if you have either the essential or the professional, you have this always. The crosstalk is its own option. The deconvolution is its own option that goes per microscopy type. So one for confocal, one for wide field, etc. Bleaching is included with the deconvolution. So if you have any deconvolution, you automatically get the bleaching. Chromatic aberration correction has its own option. A region of interest has its own region of interest are included with the analysis options. And there we have one option for the colocalization analyzer and another one for the object analyzer. And finally, there's the automation that I've been talking about that is also already included in the basic. If you don't know yet, your Huygens slices automatically enables you to just do crunch data, do batch processing, crunch your data sets, and yeah, really process a lot of data this way. Now, I'd last like to mention colocalization in 2D, because we've been talking a lot about 3D and showing 3D examples. And essentially, this whole story is also valid in 2D. There's one exception that I will now mention. So first of all, the deconvolution is not an exception. So the deconvolution also works in 2D. So Huygens deconvolution also works in 2D. You don't need 3D data for it. For example, to show that here, I've taken this image and I've deconvolved it, and I'm showing here the deconvolved image, a slice of the 3D slice. So the deconvolution knew about all the 3D information. Now here, I just took a single slice, and you can see all these blurry regions in the background that you can see here. So Huygens knew nothing about what is above or below it, but the deconvolution result very well gets rid of all the blur and just gives you a very good result also on the 2D data. And what is a tricky thing is the chromatic aberration. This is the exception that I was talking about. If you do 2D colocalization, you need to be aware of chromatic aberration in Z especially. If like in this picture, there's a shift in Z, then to capture both channels, you'd need to measure uh, um, two planes at least. If you only measure a single plane, then you will miss the information of the data that's in the other plane, like you can see from this picture. So if you have that, then you might want to consider, trying to correct for it, or at least check if you have a chromatic shift in Z before you acquire your data. Shift in XY is not a problem, so only Z you want to be aware of. The other issues are essentially fine. Crosstalk, noisy data, blurry data, bleaching, hot pixels, all of them are not a problem. And the analysis is not a problem as well. Okay, so that brings me to the end of this demo. Stay tuned. Brings me to the end of the presentation. We'll go for a 15-minute demo right after this. Tell your friends and colleagues to join as well, and send us any questions you have now or in the chat. I see there is a question in the chat, actually. Let me just look at that. Yes. If I prepared my images with AriScan option on Zeiss 880, is it better to perform deconvolution, or will it not give me any quality improvement? Yes, very good question. So Zeiss also offers their deconvolution options. We advise you to load the raw data into Huygens and make the quality test for yourself to see which you prefer most. So many of our customers really value doing Huygens deconvolution on their AriScan data. They're very satisfied with the results. So I would advise that, if possible, to get the raw data out of your AriScan microscope and load it into Huygens. Then that gives you the best quality overall. To deconvolve it again is possible, but we'd advise getting the raw data if possible. And we're happy to talk more into detail about that later, if you like. So I'd just like to mention, if you don't just like colocalization, but you also like light sheet microscopy, we have an upcoming webinar the 16th. We have a great solution there as well. So just stay tuned for that. Okay, so I just opened the Huygens Essential for the demonstration that I'll be showing you today. So the Huygens Essential recently got a bit of an update in terms of the user interface. So you can see now all the icons are collected in these menus, and I just got these nice icons that you can see here. And what you also notice is that you have a few demonstrations ready to go here on the right. So today I'm going to use the colocalization demonstration. So by just clicking this button, the data will load. Now, if you have a professional license, you also have access to this demonstration data. You just have to download it from our website. Happy to send you the link, and no problem. I see there's another question. How do you double check if deconvolution does not create artificial patterns? I only know by just looking at the images. So we have a lot of webinars addressed to this topic on how to really do a good deconvolution. And so let me just give you the answer in brief. So Huygens deconvolution is very well regularized. So just the way that the algorithm works takes care that uh, you enhance your image, but it has various ways of not over restoring your data. That is the one part of it. Another part of it is that Huygens has a lot of checks, for example, to have input data in your if you have your input data, then you want to use an appropriate SNR and appropriate pixel sizes, for example. And Huygens has very sophisticated ways of estimating these parameters and ensuring that if it's somehow the information is missing or it cannot be estimated reliably, it will give you a warning. So essentially, Huygens has been tested and really upgraded many times to give you reliable data. So it's hard to break it. Of course, if you push it, you can break it. But Huygens will warn you if you're doing things that might lead to issues. I hope that answers the question. Okay, so let me just go back to the demonstration. So this is the data that I've been showing. You're welcome. This is the data that I've been showing also in the presentation. And so these are the kinetochore complexes with the chromatin bridge. And I just made my image a little bit bigger by using here this preview. I set it to huge preview. Now, before I go on, I'd just like to point out, I've been talking a lot about also the SNR effect in the introduction. You can also find that demonstration now in Huygens. And this is the high SNR, and this is the low SNR data. So there's really 20 times difference in intensity. You can just open that in this demonstration. I will not do that now. I just wanted to point it out, and you can look at that at your leisure later. So this is a wide field data set. It has been recorded according to Nyquist. And so now I'm going through the Z slices. And what I can see is that there is a lot of blurring, what I expect from wide field, but the SNR is rather high. So this is a nice quality, nicely recorded data set. And of course, as I already alluded to, we can see chromatic aberration. On the other hand, there's no crosstalk, there's no hot pixels, and there's a tiny little bit of bleaching, but not very significant. So to restore this, I am going to use the deconvolution express. So I'm going to use this little button here at the bottom. And I'm just going to use the default deconvolution express option. So sometimes my computer struggles a little bit because it's both recording, presenting, and deconvolving. So I'm just going to accept the defaults, and these work very well, and they use the metadata, especially if you have any type of commercial system from a major manufacturer, then the metadata is read in automatically, and a one-button solution just gives you really very nice results. So here it is. So now it has been corrected for, it has been deconvolved, but not yet corrected for chromatic aberration. So let's do that next. And I'm going to do that in this menu. I can also find the hot pixel corrector, the crosstalk corrector, the bleaching corrector here. And for the chromatic aberration corrector, I'm going to use the window. The wizard has similar functionality, but goes through it step by step in a wizard. And the window just gives me all the information in one view. So I'm going to hit estimate. And then, so it's now calculating the shift based on the data itself. And it finds here a shift of 600 nanometers in Z. So that's quite significant at this resolution. It also finds a shift of 90 nanometers in X, which is also significant, and a smaller shift of 24 nanometers in Y, which is less significant, but still nice to correct for. So now we've corrected it, and now I have my fully restored data set. So I go analysis, colocalization analysis. I choose the compact. And here I have my image. Let me just make it a little bit bigger. And you see it now in blue and orange. And in your case, you might see it red and green. That's because I've enabled the colorblind friendly mode, like so. And so if I now want to calculate Pearson, I just enable Pearson here. I hit compute, and here my Pearson appears. So it's 0.489%, like we saw before. Now I can also add a ROI. So if I don't add a ROI, I take the whole image by default. But if I add a ROI, of course, I only have one cell in this image, so I don't really need to add a ROI, but I just show it to you right now. I have added this ROI, and if I hit compute again, then I will just compute it for the area which is inside of my ROI. So it's really easy to do, and you can also load ROIs from an external program if you like. So I'm just going to clear it. Then you'll notice that the Manders coefficients are both quite high. And that's because to get the effective Manders coefficient, I need to set a threshold. So in this case, for channel A, I need to set, I'm going to stick to a 5% of the maximum sort of heuristic value. And so in this case, 5% of the maximum correspond to 300 and 1,000 intensity values. And if I then hit compute again, you see that I now get the difference between M1 and M2. And we can understand that very well because the orange is always included in the blue, or most of it, 85% is. But there's a lot of blue space which isn't covered by orange, so that's only 38%. So then the Manders coefficient actually gives me the information that I, of course, can also visually see, but I can also quantify it in this way. And then by doing it this way, I can disentangle this information. Now you also see these little points appearing, and these are just a visualization aid where there are areas of high colocalization. And if I just change the threshold a little bit, then you see that these guys here in the background, they also start to appear. Okay, so just to reiterate, to get to Pearson, I just can use this tool. It has many more options, like the Manders coefficient and the region of interest. If I like, I do this for the 20 cells for each iteration 3 times. I can automate this. We'll show that in a later session. And these are the Pearson coefficients that then enter in my box plot. And if I then like, I can also use this Manders coefficient to paint a more sophisticated picture. So the last thing that I want to show you today in this demo is colocalization using object analysis. And I'm going to use the object analyzer for that. So in the analysis tab, object analyzer, and here it is. And I'm going to select the channels that I want to analyze. So that's channel number 0 in the primary segmentation group, channel number 1 in the secondary segmentation group. And you can see that the default segmentation just works quite well out of the box. And so the segmentation is okay. There's maybe a few objects that aren't completely segmented, but a lot of them are. So I can work with this. And then I want to enable different colocalization uh, uh, measurements, and I can do that in the experiment report editor. By default, Huygens does not calculate all the possible parameters because it just takes a long time. But you can do that as well. For now, we want to measure the correlation between channels. So what's the distance? What's the nearest neighbor in the other channel? We also want to measure the intersection. So which part of the object is actually also covered by the other channel. So to enable both, I go here to custom selection, and then I go edit. And then I did this before, but I go here in the nearest neighbor tab, and I just enable these three options. These are the ones I need right now. And same for the co-localization. These are the co-localization measures that I want right now. Okay, so I just click apply, and then I click OK. And now I've set what I want to analyze, and I run it to get the data that I want. So you find a lot of different parameters here. And the one that we're interested right now is this one. So it's the nearest neighbor, center of mass distance. And for example, if I click on this one, I can zoom in a bit, then here I can see both a line, I can see the object lit up, and I can see a little green line, which is the distance. So in this case, 150 nanometers. There's a question. Have you implemented the latest sum for microscopy AI-based generalized model for the segmentation in Huygens? Plan to do so soon. Yes, we have. So actually, it's this button here on the left. I will not show it as part of this demo, but I will be happy to send you the webinar. We did implement that. We also made our own improvements to make it suitable for 3D fluorescence. And this is an this is an area of active development that we're currently working on very much. It's also possible to import label images from external sources if you choose to. You're welcome. Okay, so going back, the center of mass distance. I can, for example, also make a histogram of this. And so I just need to change the range a bit because there's these outliers that I don't care about. So if I then tune the range and the bin size, I can see I have these two populations. And these are the two populations essentially that correspond to the chromatin bridges that have dissolved and the chromatin bridges that are still intact. So this one has dissolved, so it has a very small distance. And this one, so other ones like this one, oh, it's just my mouse. They have a bigger distance, so I can visualize that. And the other thing is that I can get the relative and also sort it decreasing. So decreasing, so it has the highest percentage on top. This is the percentage of the object that is co-localizing within the other channel. So these are just parameters as well as the object volume or the object sum, subject intensity. And I have all this, and I can mine this data set to find patterns and draw my biological conclusions based on that. So that brings me to the end. I just briefly like to summarize. So Huygens really provides the complete package from restoration to analysis, gives you different options to do the analysis. It can solve really complicated problems as well. We've not been covering those for sake of time, but we will go into more detail on that later. And I just want to thank you all so much for listening.